Myself, Dr. Lekshmi R Nath from Amrita Institute of Medical Science, Pharmacy, School of Pharmacy, Kerala, India. I'll be presenting the content part of that particular work, which got accepted recently in recent patents in anti-cancer drugs. I'll quickly move on to my topic. Phytochemical-based nanoformulation against hepatocellular carcinoma. Hepatocellular carcinoma, as we all know the basic concepts, it's a type of primary liver cancer and is one of the deadliest malignancies. It's third leading cause of the cancer-related death. The pathogenesis of HCC is a complex process consisting of many risk factors like liver cirrhosis, hepatitis B, hepatitis C infection, etc. The risk factor lead to chronic hepatic injury, which further progress to fibrosis, ultimately leading to irreversible liver cirrhosis and hepatocellular carcinoma. The wonderful fact about this particular treatment strategy against HCC, there currently only one treatment available as a prime line for advanced stage patient, that is sorafenib, an oral multi-kinase inhibitor. Several incidents of adverse reactions being reported on the use of sorafenib. The number of HCC cases is rising and there is still no efficient treatment available. An approach that has a better therapeutic effect with well-tolerated and minimal adverse effect is highly essential. Coming to the topic of interest is natural products. What is the importance of these particular natural products in this particular treatment scenario? Natural products are prone to be effective cytotoxic agent. They are found to affect stages of initiation, promotion, progression in cancers, including HCC. And microorganisms, marine organisms, and plant extracts show profound biological activities. And phytochemicals are promising candidates for cancer therapy as they have less systemic toxicities, more biocompatibility, and more eco-friendly approach. But the major hurdle in the translation of this particular part into an effective drug is their bioavailability issue and pharmacokinetic limitations. The pharmacokinetic properties of molecule affect its solubility, absorption, permeability, stability, and physiological pH. These can be overcome with an effective drug delivery system that is capable of targeting the molecules into the site of action. Then here comes the relevance of nanomedicine. Nanomedicine has numerous benefits such as increased bioavailability, drug efficacy, targeting, functionalization, etc. compared with the traditional drug. Nanoparticles have increased strength, durability, performance, flexibility, and unique physicochemical properties that can be utilized in various treatments. The nano drug encapsulates the biological agent in a nanocarrier. They also provide added advantages of limited off-target adverse effects and protection of drug from degradation. Due to nanoscience, the drug uptake will be rapid and easy, ensuring desired targeted drug delivery. The biological effects of the natural compounds can be increased when given as a nanoformulation by improving pharmacokinetic and pharmacodynamic properties. Then what is the relevance of green synthesis of metallic nanoparticles? Here we are focusing on the metallic nanoparticles. Even though we have n number of formulations, then here the chemical and physical process used for the synthesis of nanoparticles are not economically feasible and produced toxic byproducts. More eco-friendly and less toxic alternative is always required. The green synthesized nanoparticles has a predominant advantages of conventional nanoparticles like economical, efficient, and simpler synthesis, and eco-friendly approach has led lesser systemic toxicities and more compatible. The green synthesis method of nanoparticle synthesis adopted that uses phytoconstituents and microbial extracts. Usually, the nanoparticles are normally synthesized by mainly two methods. We can call it as bottom-up method and top-down approach. In bottom-up method, the assembly of the material begins at the nanoscale, such as from atom or molecule level, and then made to form large molecule. In top-down approach, starts from a bulky molecule. It involves cutting down of the complex molecule into small molecules and creating smaller crystal from a bulky molecule. There are various techniques used in this particular methodology like mechanical milling, spurting, etc. Then I'll not be discussing much detail into that as my presentation is restricted to 15 minutes. Then we'll be discussing about its mechanism of action. When you search the mechanism of action of any particular kind of the nano formulation, we'll not get a much detailed information on the particular key regulatory mechanism. Key regulatory mechanism in the sense, usually we'll call it as the cell death mechanism, as apoptosis, autophagy, all these kind of terms. The regulatory mechanism means actually the signaling pathway regulated by the particular kind of formulation. Then usually that limitation is always there. Like mechanism of action of green synthesized metallic nanoparticle is not fully understood. The nanoparticles increase the level of reactive oxygen species within the cells, leading to DNA damage, then membrane destabilization and mitochondrial dysfunction. It also induces lipid peroxidation, modulates gene expression, ultimately cause cell lysis. The cytotoxic effects are usually brought out by the mechanism of, as I mentioned earlier, like apoptosis, caspase-mediated transduction, etc. So here you can see a few examples, as it is the topic of green synthesized nanoparticles, collected the information from our article. Like these are some of the plants like which being fabricated with a particular kind of metallic nanoparticles, and all these kind of works, it's already up to the level of in vitro analysis using the cell line, Hep-G2 cell line. Hep-G2 cell line is a liver cancer cell line. Then coming to the second topic, like using the magnetic oxide nanoparticles, here also we can see the research only comes up to the level of in vitro studies. Then regarding the other kind of nanoparticles, metallic nanoparticles like tin oxide or silver peroxide, etc., the same case only. Then these are some of the clinical relevance of nanoparticles. We'll be talking about the green synthesized nanoparticles. This table pick what extent the concept of nanoparticles, the conventional nanoparticles, reach to the level of a certain clinical trials only, but no drug is available as a clinically used drug. Then as we mentioned earlier, there are a lot of challenges because we have mentioned many reports, but all these reports are up to the level of in vitro studies. That is a major limitation where we focus now the translational gap between animal models and humans. Then biological materials present on the medium such as amino acids, proteins, etc. can adhere to the surface of the metallic nanoparticles, and it can change in vivo biological activity. That may be the reason for the limited number of the preclinical evidence. Like then to conclude, the green nano biotechnology can offer notable advantage over conventional method in terms of improved stability, drug carrying capacity, site specificity, feasibility to apply different routes of administration with lesser systemic toxicities. The green synthesized metallic nanoparticles can thereby be a highly beneficial strategy against HCC if properly validated with preclinical and clinical studies. This is all about my conclusion. I would like to thank my PhD students and informed students who are constantly working on this particular aspect. So as my presentation is limited to 15 minutes, so I could present only the content of that particular thing which we have published recently. Thank you.